No, I think I skipped put it because I asked him. Oh, yes. It comes before variable videos.
Yeah, put it in. Yeah.
It's in.
We're good?
Yeah.
No?
We're
So
being
it's already
recorded,
recording?
Doctor.
Hello out there. Okay.
No video though, at least that.
Discuss.
Okay, so today's lecture for a huge group of neurology residents and students is sort of the introductory to the molecular biology of neuroscience. In that we're going to look a little bit at the different neurotransmitters. This is kind of a very productive field in terms of pharmacologic interventions, in terms of sort of understanding how nerves work, and it's generated sort of a lot of literature over the years. It's also generated a number of Nobel Prizes and other things like that because of the complexities of the systems involved. There's no way to cover all of this, and most of you have probably covered most of this in either basic neuroscience, medical school, or if you're able to take a graduate course or undergraduate neuroscience, you probably covered this in much greater detail than we'll be able to do today. So this is kind of a survey. And the basic idea of neurotransmitters is that they are the chemical link between two neurons. They provide us a way of getting a signal from one neuron into the next using a chemical signal that's released on the presynaptic neuron and then goes across the axon plate over to the postsynaptic neuron. Very basic stuff. The different molecules that are involved is basically what this talk is going to be about. In this diagram here, there's a couple of things to take note of. There's some systems in place to make sure that the system all works efficiently and that you don't waste a lot of biochemical energy just regenerating these neurotransmitters. So there's a reuptake system for the majority of the neurotransmitters, at least the small molecule ones. And on the receptor side, there's also intake of these. And there is a pathway to get some of them, not all of them, back over to the presynaptic side. It's a little complicated. It has to be transformed into the precursor and then sent back over. The mechanism of transmission, basically signaling energy, neurotransmitters packaged by the nerve is put into vesicles at the terminals. This is pretty labor intensive in that some of these vesicles have to travel all the way from the cell body out. Some of the packaging does occur at the terminal, and that makes things a lot more efficient. Then, once you have the vesicles of the appropriate transmitter type, they are released with the appropriate signal, meaning depolarization of the presynaptic neuron. And then they fuse with the membrane, release their contents, exocytosis as it's called, and the contents go out into the cleft between cells. The transmitters then degraded or transported back into the cell. And principles, that basic mechanism is basically how most transmission is thought to occur. There have been some developments in the last few years where there's sort of things that go around the system without using vesicles. There's other new players that are starting to come into play. Categories of neurotransmitters, these are basic categories. Acetylcholine is a big one. It is, I guess, the second most common neurotransmitter after GABA. And then there are what are called amino acid transmitters. Glycine, GABA is an amino acid transmitter. There are others. Biogenic amines include serotonin, norepinephrine, dopamine, and purines are used sometimes. Peptides are also used in substance P. And endocannabinoids and nitric oxide are also used as neurotransmitters. Nitric oxide is one that actually does not use the synaptic or the vesicular release. Now I'm not sure, maybe one of you guys knows endocannabinoids I don't know if they have vesicles or not. That's something I probably should explore. So acetylcholine, this is the classic transmission that was first investigated with squid giant axons and then onwards from there. And this is just to show sort of insight to the final changes. The acetylcholine is formed from acetyl-CoA and choline. And they show it here at the terminal. I presume most of it happens at that location. The vesicles themselves, the package of membrane probably has to transport down down the axon all the way to the end, and then it gets filled at the end. Okay, so nicotinic receptors are one of the two grand categories of targets for this, and these are the ones that will move your muscles and do that kind of stuff. Muscarinic, so the other ones I'd like to have a diagram of that. These are relatively complex receptors as these go. They have, I think they have 4 components that get structured in, actually I think it's 5, anyway. But, or it's the 5 transmembrane domains, and there's 4 of these get packed together. Now the ones that get used varies a little bit during time in development. So if you have one that's slightly more likely to get inappropriately excited, like an epilepsy, that may change later on in life. And the time points for change are typically sort of pre-adolescence, about the time of puberty. Then there's another time point at the end of puberty, about 17, 18, another one around 20, and the last one 27. And so if you have a bad receptor, you can actually sort of graduate out of it and mature into more normal receptors. Acetylcholine as a transmitter is the principal transmitter at neuromuscular junction. That's in the nicotinic receptors. These are ion channels. Muscarinic receptors are the other target for acetylcholine. They're metabotropic, so they're G protein coupled. When the acetylcholine binds, it does actually, sorry, acetylcholine is degraded by acetylcholine esterase, which is all scattered throughout the area, and the choline itself is what's recycled. Now, the receptor itself, because it's so intimately involved with movement, is, you could say it's a target, but that assumes some kind of intelligent being figuring this out. Evolution has found that chemicals can bind to those receptors, cause paralysis, and then certain organisms have produced these toxins, and that's where we get the neurotoxin and other toxins that basically will freeze a prey or selected animals to have the toxins in them. It is a heteropentamer receptor, so I guess it is 5 components as well. Five loops to the structure. Okay, the toxins that are involved, organophosphates inhibit acetylcholine esterase, so you'll get sort of overstimulation of acetylcholine, you'll get flushing, you'll get sort of excessive secretions. Those are the kinds of symptoms you might get with that type of toxin exposure. Alpha-bromneurotoxin is a nicotinic specific toxin, and I believe that one's from, is that one from the frog, I think? No. Chronotoxin, chronotoxins also block receptors. These are just different examples of different toxins. And then actually nicotine itself and betel nuts have agonists as part of their composition. So nicotine is at least a partial agonist. Botox is basically blocks synaptic release, so it works on the presynaptic side of this junction. And atropine works not on the nicotinics, but rather on the muscarinics. And so you'll get more autonomic type responses. And this is why it was used to dilate the pupils way back. Scopolamine also blocks muscarinics. It's anticholinergic. Myasthenia gravis, sort of a little more clinical correlation. The antibodies that are involved with myasthenia gravis are, there's several different types. There's a binding, blocking, and a modulating antibody that are categorized um, specifically for this disorder. Receptors are removed from the cell surface after the antibody binds. And the effect is that basically there's not as many targets for the acetylcholine, so it doesn't have as much impact on the postsynaptic receptor. This is partly why when you do repetitive activities, you're sort of stimulating the receptors, the antibodies are around, and you end up bringing more of them in. They get degraded, and it takes a while to replace them. Use-dependent weakness is sort of the cardinal feature of this disease, and the duration, enhancing duration of acetylcholine helps. That's why the mestinon, which actually blocks the acetylcholine esterase, helps a bit. Myasthenia gravis is associated with thymoma. The connection, I think, is still a little ambiguous, but it's something we look for because of frequent comorbidity. Okay, glutamate. This is also regenerated at the synaptic terminal, and this is actually visible as opposed to the previous one. What's going on here is that the glutamate goes out, it goes across to the postsynaptic neuron, stimulates the glutamatergic receptors, and then some of it's going to sort of get out of the gap and go into nearby glial cells. In the glial cells, the glutamate will be transformed back to glutamine and then transported out of the cell and retaken up by the terminal again and reprocessed. So the core amino group is, and it's sort of its constituents is recycled. That saves a lot of energy. It makes it a little bit more efficient. Again, the vesicles themselves have to be constructed at some point. So basic diagram there. Glutamate is the primary activator in the brain. It's an amino acid. It has receptors that are, there's three different larger categories of receptors. There's AMPA receptors, which are what we think of sort of as typical excitatory channels. Kainate is also, I think, somewhat excitatory, and NMDA is atypical. It has a very unique mechanism, and there's several cofactors involved with that. Those are all ion channels that work different ways. The metabotropic receptors, there's actually more of those, in that there are, I think, at least 7. I think it's gone up from there, probably 8 or 9 at this point. They're G protein linked, and what exactly they do has been an active target for research and development of a couple of medicines over the last 10 years or so. It's not one of those medicines, I think, is related to insomnia, and that one that came out a couple of years ago. There hasn't been a whole lot else yet with metabotropics. NMDA is different because it's not strictly an ion channel. There's cofactors, including magnesium, that are involved. It is a target for disruption in that we block it with NMDA antagonists in dementia to sort of slow down overstimulation. It works to sort of lubricate the brain, make things a little more efficient. But it's a temporizing measure. It doesn't have any impact on actual dementia or plaques or anything. Glutamate, as we saw on the slide before, that is removed by the glial cells and recycled, and it's transported back to the nerve.
So NMDA usually is a sodium channel, or?
I think it's a calcium channel. Yeah. Here's the NMDA, and I don't think I can blow this up. But let's see, there's calcium. Calcium's right there. Sodium, both. We got both.
Okay.
Sodium and calcium. There's the glycine binding site. There's the magnesium that sits right here at the top of the pore and sort of blocks things. And when the channel opens, the magnesium pops off. So if you keep the magnesium from popping off, you kind of block the channel. Very much involved with memory and training as well. So that's one of the reasons overstimulation of this is sometimes a problem.
Yeah, we have a secret.
You know,
We have a secret.
could be. Well, then there's there's an antibody that targets this that causes encephalitis also. So it's sort of like the idea of the nice thing. Okay, GABA. So GABA is probably the most prevalent single neurotransmitter in the brain. It's something like 80% of nerve cells are set up to be inhibitory, and the primary inhibitory neurotransmitter is GABA. It's also got a biochemistry. It's also recycled, similar to the other two, all done at close to the terminal. A principal inhibitory neurotransmitter in the brain, about 50, I thought it was more than that actually, but 50% of neurons are GABAergic. I guess the word glycine is also inhibitory. Glutamate is decarboxylated to make GABA. So the same molecule that is excitatory is actually reused to make GABA. This is pretty efficient stuff. There's a special transporter into the vesicle. I don't remember the full set of that acronym right there, but vesicular something. We'll look it up quickly. It's stopped by basically the action of the GABA, slowing down transmission or slowing down certain excitatory pathways, is actually stopped by the transporter itself rather than degradation. In some receptor ion channels, some receptors, the GABA is on ion channels. There's also metabotropic GABA receptors. Again, okay. So this would be because it's on the vesicle rather than the membrane, and an inhibitory amino acid is the GABA, and then transporter. Is it transporter or amino acid? What's the extra T? Okay. Trying to figure out why there's a second. Oh, I got it. I got a typo. I'm sorry. Okay. Okay, and this is the GABA receptor. The GABA receptor is important, not only because of the basic function, but because it's the target or the mechanism for a number of medications and substances of abuse. So the barbiturates acts through this. Benzodiazepines the site is up towards the outside. GABA itself is close by, but not the same place. So you could block GABA, but not block the benzo, vice versa. And the whole structure, alcohol itself, I think actually has more of an effect on the membrane that somehow changes the conformation of the proteins. So this is the target for benzos, alcohol, barbiturates, the whole shebang. And that's why we can sometimes get away with treating or covering somebody who's going through some kind of withdrawal, i.e. overactivation, underactivation of the GABA channels with a cross agent. So you can use a benzo to stop somebody from withdrawing from barbiturates. You would not be able to do the same thing for a narcotic because it's a separate receptor. GABA targets, GABA A allows chloride current, which is an inhibitory current. Benzodiazepines are agonists. GABA A, as we saw from the diagram, it's not quite the same site. Barbiturates are also agonists. Alcohol is a modulator through the membrane. Picrotoxin is a potent antagonist. And this is one of the old models of seizures, was to basically apply picrotoxin to the surface of the brain. Wicked stuff. Without the GABA, everybody seizes. So very severe lesions that are left with that, but they generate seizures later on. So it's a model system. GABA B activates calcium channels, which are calcium going in, chloride coming out. also both those mechanisms are inhibitory.
So our brain is having more inhibitory process going on
Yeah.
at the same time.
Most of the time. Yeah. Okay, and let's see, this is glycine, and another inhibitor, and it's got the same. If you look carefully, I don't know if you guys can see this.
It's not a typo for you.
I have it here
Yeah,
in the.
I don't know. Maybe the author of the book got it all wrong. It's okay.
They have it here in it's a study, vesicular inhibitor amino acid transporter expressed in GABA containing astrocytes.
Yeah, no, your interpretation, but nevertheless, it's at least I was consistent with the typo. Okay, so the transporters there, it transports both. It's the same transporter, and the same idea. The inhibitor goes out, it goes to the cleft, gets I guess it goes back and becomes sort of degraded, and then glycine goes across.
It's
The acronym
abbreviated
is
of
correct.
both. It's abbreviated of both. Here it's
two different.
VIH. No, it says
No, it's the same.
vesicular inhibitory amino acid transporter, and then here it's also vesicular inhibitory amino acid transporter. So they do it
both of them is
VIAAT or VIATT.
Yeah.
So both are correct.
I'm wondering if they there's a little history behind the transporters here, and I'm not sure the particular thing. I think it was found. I want to say it was found in Duke, but or at least characterized as Duke. So I'm not sure if it's a language thing. Duke speaks English.
Yeah.
So glycine inhibitory mechanism, particularly in spinal cord, it's a target for strychnine, which is a toxin. It is cleaned up by transport. Transporter mutations cause hyperglycemia, can cause seizures and eventually mental retardation. So that's actually, I think that's one of the things they screen for, or they used to screen for newborns, is a glycemia, or hyperglycemia. Metabotropic receptors. So this is kind of a, let's see, you count 1, 2, 3, 4, 5, 6. Looks like 6 trans. No, it doesn't. It's got to be 7. So 7 transmembrane domains. The G coupled protein part is down here, and the outside the cell here. This would be where the receptor would bind to the ligand or the neurotransmitter. The mechanisms behind this are complex at sort of the basic level. This is breaking things down a little bit differently, looking at some of the biogenic amines and going in this direction. These particular molecules obviously are involved in a number of basic functions of the brain, but particularly sort of psychiatric homeostasis, if you want. So dopamine, norepinephrine, epinephrine, as well as serotonin. These are huge targets for antidepressants and for, if you consider, I guess you don't consider it psychiatric, but for Parkinson's disease, and the treatment of the same. All of these are used as transmitters. They're all recycled as well, just like the other ones, the amino acid recycled systems. And the systems where these come from, are generated and returned to, are fairly restricted. So the dopamine is mostly the midbrain. There's another smaller nucleus, norepinephrine, locus coeruleus, and then the serotonin, epinephrine, in most places. And the serotonin is the RAF-A nucleus. Histamine, I'm not even sure where that comes from specifically, nor much about its function. Okay, so this is to show some of the connections that are ongoing. The actual dopamine, this is an example of the dopaminergic system, comes from this area right here and another nubbin somewhere. I think it's up over here. And then projects forward into the brain, all around the brain, controlling premotor, motor, a lot of different areas. And then there's some projections that go back around, circle back into the brainstem. So it's a relatively elaborated network, and when the network stops functioning as it should, which happens to everybody with time, with Parkinson's it happens earlier, and you start developing some abnormalities of control of movement. The actual coordination centers, if you will, is in the cerebellum to a large extent, though there's some cortical control as well. The oscillators for movement seem to be directly connected to this sort of network, and that's why we see a lot of tremor as well as rigidity with Parkinson's disease. There's other ways of breaking this network apart. This is just to show sort of the projections from the substantia nigra. The dopamine. Dopamine is dopa decarboxylase is a rate limiting enzyme. So if you want to slow down dopamine production, that would be a great target. That's not a particularly appropriate target for neurology because that's generally not our aim. It's released after it's been synthesized, and the receptors are all metabotropic. There's no ionic chains for dopamine. It's degraded by two enzymes, two different enzymes, both of which have been used medically. So COMT and MA, catecholamine O methyltransferase and monoamine oxidase are the two mechanisms. I think COMT is 20% and there's 80% that I put up in myself. There's also rapidly recycled by the transporter. Here, timing is important because the enzymes are actually not so efficient and not extensively present, and recycling tends to be pretty hefty. Dopamine is essential for control of movement. It also has a substantial role in pleasure and addiction. So this is part of its role in the brain. Overactivity and hallucinations are potential problems with overstimulation of the dopamine. One of the other side effects we've gotten as we've tried to muck with the system and enhance people's dopaminergic activity is compulsive behaviors, gambling, things like that tend to be potential side effects with increased dopamine. Repetitive actions are also noted, and it has an anti-nausea effect in the brain. Dopamine in the gut has a pro-nausea effect, so it depends on where it is. This is actually, that little thing there is the reason we have two components to cinnamon. So the carbidopa blocks the dopamine receptors in the gut so that the regular L-dopa can, or the levodopa can go up to the brain and bypass it. Okay, this is the locus coeruleus for norepinephrine. Similar projection pathways, but starting in a slightly different location, cardiac. And it's just to give you an idea how widespread the networks are. Norepinephrine, alpha adrenergic receptors is where it acts. A1 is a slow depolarization that I guess it blocks the potassium channel, so that would end up hyperpolarizing the cell a little bit. And A2 hyperpolarizes as well. Beta adrenergic receptor targets are present for many medications, like blood pressure medicines as well. And norepinephrine transporters blocked by amphetamine. So if you give somebody amphetamine, basically they're not recycling the norepinephrine. It stays out and eventually gets degraded by COMT and monoamine oxidase. So that's the effect of amphetamines, to have too much norepinephrine around. Amphetamine also, to some extent, blocks dopamine uptake.
When you use the COMT and inhibitor, it does not affect the.
I think it's just less of the norepinephrine in the brain in comparison to the dopamine. But that's good. It's a good point. It's a good point. I'm not sure if there's a direct impact. If anybody knows of a direct impact on blood pressure medicines, I don't.
I'm not aware.
Yeah. This is epinephrine. This is epinephrine itself. Seminal release process is reloaded by the vesicular monoamine transporter, which is different from the other one with the incorrect acronym. There's no transporter for reuptake though. So it's loaded on, but we don't really take it back up. I think it sort of diffuses and maybe degrades. Acts on alpha and beta adrenergics. And this is the epinephrine that you think of when you get sort of a fight or flight mechanism. It comes from a lot of places in the body, and it's from the sympathetic nervous system. Okay, now this one's hard to read, sorry. Histamine. Okay, so histamine is produced from histidine. It's packaged by the VMAT. It's similar to the last one. There's no reuptake, so it basically diffuses. Receptors are metabotropic. This is a common theme for the amines, the neurogenic amines. And histamine is involved in arousal, or this waking up, thinking of anything sexual at this point, I don't think, and allergies. Antihistamines can cross to the brain. I think I probably put this little point in here because that in some ways arousal can be thought of as a disinhibition or an active process. The process of arousal is involved with seizures. Antihistamines actually make seizures more likely. So this is one of the reasons we try to avoid them for seizure patients. Can't do it completely, but we try to minimize it. And this is Raphe, so serotonin. Go back to the diagram. So a lot of these things are coming from midbrain or pons. This is the dorsal Raphe nucleus. The serotonergic system that follows similar pathways to the others. There's also it's a little tiny nucleus. I think that's the nucleus of Meinen, I remember that. And that's down in the pons. So tryptophan hydroxylase is rate limiting for this one. Again, might be a potential target if you're trying to reduce serotonin. In general, psychiatry would be more acting on this particular compound, and you wouldn't want to decrease it with anything, you want to boost it. BMAT takes it into vesicles, and serotonin transporter is the reuptake pump, very efficient, sits on the presynaptic neurons, and that's the target for all the SSRIs. Receptors are mostly metabotropic, and there is one ionotropic, which is 5-HT3. There are lots and lots of receptors for most of these. Widespread effects include behavior control, moods, and satiety, so getting satisfaction complete. So that's kind of similar to the dopamine pleasure type response, but it's a different transmitter. ATP is also a co-transmitter released with other transmitters. It is excitatory, and it acts at purinergic receptors and ionotropic and metabotropics. So it can be either an ion channel or it can modify things through G protein coupling system. It has some role in mechanosensation in nerve. That's going to be probably another topic later on. Peptides. So these are more than one amino acid. We're talking multiple amino acids thrown together. We're talking peptides. They're packed into vesicles and then degraded after release. So we got a couple of them. There's substance P, CCK, vasoactive, opioid peptides, enkephalins, endorphins. So those are actually peptides. And the pituitary peptides, which are basically control mechanisms for the endocrine system. Hypothalamic releasing agents include TRH, LHRH, somatostatin. Dopamine is controlling some of these. So anti-dopaminergics have some role in controlling the release of some of the other peptides. Others include angiotensin and neuropeptide 1. Endocannabinoids, nitric oxide. So endocannabinoids are lipids, so that sort of answers the question about do they have vesicles? Probably not. They diffuse through membranes. and they inhibit feedback between postsynaptic cells and presynaptic cells, so they slow down transmission. They seem to reduce GABA release. If they really reduce GABA release a lot, we could get some excitation. But in point of fact, we're trying to use these to control seizures. So maybe it's doing it just right. Nitric oxide is diffusible and it has a localized effect. It's technically a gas, but it goes through liquids and does diffuse through membranes. It has a mechanism that's similar to metabotropic effects. What does that mean? It means it's going to act through a cascade on the postsynaptic side rather than through a receptor. Neurotransmitters and receptors, there's many different agents, many targets, there's a whole thing, a bunch of things going on at once. I partly put in those diagrams, although they were a little too small, some of them, to show you how extensive things are, realizing that each of those is a different system. It gets coordinated, there's feedback between the systems. Things get very complicated in a normal individual when you start to say, well, how do people vary? And things get very, very complex. So we're still struggling to try to work with this, and typically approaches to try to treat things are kind of you experiment, see what happens, and hopefully things work out well. That said, these compounds that work through the neurotransmitters are multiple and have treatment of depression, hypertension, Parkinson's disease. There are a lot of these agents. The pipeline for a lot of these medicines is now kind of drying up because the useful compounds have pretty much been sorted through. And the next direction will probably be more peptides. The next targets. Some reference. That's the textbook we're using for this course. Okay. Any questions, thoughts? Other abbreviations?
It does have adverse effect of hypotension.
Which one? Oh, which one?
That's a largely animal inhibitor. But
It
then
does
dopamine itself is a suppressor, so
Yeah.
that alone is.
So yeah, if you inhibit it and you get more dopamine, yeah, why would you get hypotension?
Yeah, so it is. So there could be from norepinephrine. No, that should be more.
Well, there's yeah.
The initial dopamine, low-dose dopamine.
No, we're talking about why in MMO inhibitor, it might be affecting the degradation of norepinephrine as well. So whether there's an effect on that, but then the blood pressure is actually the adverse events.
Maybe alpha twos. I don't know. Because if there's extra alpha 2 stimulation, you probably get.
So,
less
I don't know
alpha-2
the short answer, but the mechanism that I'm looking. So we talk about it. If you have an MAO inhibitor, and then you have hypertension.
Yeah.
So mechanism, my fairy tale in my head is that you increase dopamine in the blood a little bit, and then you open up the renal vasculature. Because that's a known. We always say you can't taper dopamine. When you use dopamine, you can't taper it to zero. You have to jump off somewhere else below 5 because you open the renal vasculature and make the patient hypertension. So a little increase in dopamine and then NLB inhibition may open up the renal vasculature and make the patient sick. I don't know if that's true.
No, that's worth thinking about.
Yeah.
Okay.
Oh, yeah. I'll
Stop
just,
recording.
yeah, just press stop.